Hi everyone, today we're going to learn about DNA replication. So DNA replication is when DNA has to make a copy of itself. So if you think back to last semester, we left off with learning about mitosis, or the process in which your cells divide and multiply. And one of the things that has to happen during mitosis is your DNA has to be copied, right? You have to have new DNA in your new cells. So we call that process DNA replication. And your cells replicate or make copies of its DNA by using something called like this semi-conservative approach. So what happens is you have this original strand of DNA, okay? And as we learned the other day, that these bases are held together by hydrogen bonds. So then these hydrogen bonds get broken, and then new hydrogen bases get put in on each side of the DNA. Okay? And so when your DNA copies itself, one strand of DNA turns into two strands of DNA. And you can see that on your new strands of DNA, one side of it is your old original strand, and the other side of it is that new replicated strand. Okay. Same thing over here. Here's your original strand of DNA, and then this is your new replicated strand of DNA. Okay. So we call this semi-conservative because half of it is old and half of it is new. All right, so here's some big players, if you will, when it comes to DNA replication. DNA helicase is an enzyme that comes in, and it's going to break the hydrogen bonds between the base pairs. So we say it like unwinds or unzips the DNA. And then DNA polymerase is an enzyme that comes in and it adds the complementary base pairs and it proofreads the DNA. Okay. And when we say complementary base pairs, we are talking about Chargas rule, where A pairs with T and C pairs with G. So here's a picture that diagrams what happens during DNA replication. Okay. So you could think of this enzyme as helicase. So it's going to come on through, and it's going to break these hydrogen bonds, and it's going to separate our strands of DNA. And then DNA polymerase is going to go through, and it's going to read the original strands of DNA, and it's going to place those complementary base pairs on. As it does this, it's also going to proofread and make sure that it's actually pairing, again, A with T, G with C. Okay. Same thing down here. Okay. So this strand also gets read, okay. And DNA polymerase goes through, and it's going to read those base pairs, and it's going to put on the complementary pairs. In a little bit, you're going to watch an Amoeba Sisters video that also shows you how DNA replication works. Please watch the video. It is more complex than this. So one strand, polymerase is going to move this side and go from left to right on the DNA. On the other strand, polymerase is going to move from right to left on the DNA. Okay. So please make sure you're paying attention during the Amoeba Sisters video so you can watch their nice animation of how DNA replication actually occurs. All right, so now we are going to practice replicating our DNA strand. And again, to do this, we're just going to use Chargas rule. So if you need to write this down to help you, you can write it down. So A pairs with T, G pairs with C. All right, so here we have 3' to 5', and I give you the lists of our base pairs of DNA. Notice we keep them in 3s. Again, when I get back, we'll talk about why we keep them in 3s. Okay. So when DNA replicates, it moves in the opposite direction. So the replicated strand is going to go 5'. And then I'm just going to look. So I have A. So the complementary letter to A is T. And the next I have T. So complementary to T is A. And again, T is A. Then I have G. So the complementary letter is C. And then C's complementary letter is G. And G is C. On my next set, ATC, my complementary letters would be T, A, G. Then GGA, the complementary letters would be C, C, T. And then CGT is GCA. And then finally, we're going opposite 5' to 3'. Okay, let's do it one more time. So here's a new strand of DNA. I want you to try this one on your own. So pause this video. You write down what you think the replicated strand is going to be. And then when you're finished, go ahead and play this video and you can check your answers. Okay, so the replicated strand is going to go 5' and then A, T, A, C, T, G, A, G, C, C, G, 3'.